Ready!
Hey everyone, we are the Study Buddies. Welcome back to our channel. In today's video of Biology Edition, we will be learning about another new subtopic, which is prokaryotic cells. So basically, all life on Earth are classified into three domains. This phylogeny or evolutionary tree shows the three divisions in life. The single line below here is where all the three domains of life came from, which what we call as the last universal common ancestor, or LUCA, LUCA. The three domains are namely Eubacteria, Archaea and Eukarya. Just because all life on Earth came from the same last universal common ancestor, all life shares some common characteristics, such as being composed of cells, containing DNA and RNA, have a phospholipid-based cell membranes, and they use protein catalysts such as enzymes to speed up biochemical reactions. For a long time, bacteria in the domain Eubacteria and archaeans in the domain Archaea were thought to be closely related because of their similar shape and sizes. Comparisons in DNA and RNA sequences show they are distinctly different from each other. In fact, archaeans are much closely related to eukaryotes instead. Now, if we classify them in terms of the type of cells they have, fundamentally, there are two types of cells that exist in nature. One is the prokaryotic cells, and another is eukaryotic cells. Prokaryotic cells lack a nucleus, while eukaryotic cells have a nucleus. Generally, most organisms in the domains Eubacteria and Archaea have prokaryotic cells, while eukaryotic cells can be found in organisms in the domain Eukaryota. Firstly, let's look at the group Prokaryotes. This group is one of the most abundant and diverse life forms on Earth. Although they are structurally less complicated than eukaryotes or eukaryotic cells, their metabolic capabilities as a group far exceed those of eukaryotes. Prokaryotes are an extremely successful group of organisms whose evolutionary history dates back to the first cells on Earth. Structurally, there are various shapes and sizes of prokaryotic cells. For example, coccus ones are shaped like tiny balls or beads, bacillus are rod-like shaped, and etc. Generally, all of them are quite small with an average size of 1.1 to 1.5 micrometer. The generalized structure of a bacteria or prokaryotic cell can be divided into three main parts, which are the envelope, cytoplasm, and external structures. Firstly, let's look at the cell's envelope. The cell's envelope consists of three main parts, which are the plasma membrane, the cell wall, and glycocalyx. Firstly, the plasma membrane. The plasma membrane is what we have learned in the previous video before. It's made up of a phospholipid bilayer. Its main function is to regulate the entry and exit of substances into and out of the cell. Some cells would have structures called mesosomes or like internal pouches. These mesosomes are to increase the surface area for enzyme attachment that carry out metabolic activities in the cell. Next, we have the cell wall around the cell that maintains the shape of the cells. This cell wall of prokaryotic cells are unique because they have a complex disaccharide called peptidoglycan covered around it. With the help of biotechnology, scientists have found antibiotics that target these peptidoglycan in the bacteria in order to destroy them. The final component in a cell's envelope is called the glycocalyx. In some bacteria, they have glycocalyx, a layer of polysaccharide that lies outside the cell wall. Its main function is to aid against drying out, helps bacteria resist host's immune system, and allows attachment to any surface. In a slimy state, the glycocalyx is easily washed off, but when hardened, a glycocalyx is called a capsule that encloses the bacteria and is not easily washed off. The second structure in a prokaryotic cell is called the cytoplasm. A cytoplasm, as you may have known earlier, is a semi-fluid solution composed of water and organic molecules. It is more like a watery environment where all the chemical reactions takes place. While prokaryotes lack membrane-bound nucleus, so where does their DNA or RNA are located? The DNA is located in a region called the nucleoid, where the DNA strands are just coiled up into a single chromosome. Many prokaryotes also have extra chromosomal pieces of circular DNA called the plasmids. They are generally like mini trucks carrying DNA to the outer side of the cell. These plasmids provide antibacterial resistance too. Prokaryotic cells also have structures called ribosomes scattered in their cytoplasm. Ribosomes' function is the same in eukaryotes too, where they synthesize the proteins needed by the cell. Simply remember ribosomes as the site of protein synthesis. Ribosomes also have larger and smaller subunits, which we will learn more in our future videos. Next, we have the external structures. When we look at some of the external structures in prokaryotes, first we have what is called a flagella or this tail-like structure over here. Most bacteria cannot move by extending their cytoplasm like what amoeba does by extending its pseudopodiums. So, in order to locomote, they use this flagellum to move. The flagellum rotates 360 degrees, causing the prokaryote to propagate in different directions. Next, we have the fimbriae or these hair-like structures. They are simply for attachment purposes. Lastly, we have the conjugation pili. Prokaryotes generally reproduce asexually by binary fission, but these conjugation pili are used to exchange DNA between the prokaryotes. Even though I said earlier before that prokaryotes and eukaryotes are distinct from each other, but evidence suggests that eukaryotes evolved from prokaryotes in the earlier days. We know the main difference between prokaryotes and eukaryotes is the nucleus. This distinguishing characteristic in eukaryotes, which is the nucleus, is believed to have evolved due to the invagination of plasma membrane in the prokaryotes. During the invagination of plasma membrane, the cell membrane folds onto itself, enclosing the DNA in the cytoplasm. This forms the nucleus and also leads to the formation of endomembrane system called the rough endoplasmic reticulum and smooth endoplasmic reticulum. Some other evidences suggest that energy organelles in eukaryotes, such as mitochondria, were originally aerobic heterotrophic bacteria, and chloroplasts were originally cyanobacteria. The eukaryotes might have benefited from the ability to utilize oxygen or synthesize food on its own, while the prokaryote got a safe place to be in. This entire relationship between eukaryotes and prokaryotes is explained via the endosymbiotic theory. Endosymbiosis occurs when a host engulfs a smaller cell and is able to use it for special functions. In this picture, it is shown that the eukaryotic cell is the host, while the aerobic or prokaryotic cell is the smaller cell. So, what is believed to have happened is that the bigger eukaryotic cell engulfed the smaller prokaryotic cell and used it for its own needs, such as utilizing oxygen and making its own food. So, this is how eukaryotes were derived from the prokaryotic cells. And with that being said, this will be the end of this video about prokaryotic cell and little bit on the endosymbiotic theory. Thank you everyone for watching this video, and I really hope you learned something new today. If you like this content and want more of it, do like this video, share it with your friends and family, and subscribe to our channel. Don't forget to hit the bell icon as well. If you have any doubts regarding any subjects, do comment your question in the comment section and we will help you to answer it. Thank you everyone and take care. Goodbye.